guys, this is Mrs. Krause, and this is a video about the second half of the Unit 8 Lesson 1 notes on Mendel and basic crosses. In class, we've already covered about half of these notes up until where it starts talking about test crosses. So if you could go ahead and flip in your notes to where it begins discussing test crosses, that would be lovely. And we'll get started. So a test cross is a genetic cross where we do not know the genotype of one of our parents. We know that that parent shows the dominant physical trait, but that parent could have one of two genotypes. Could have the homozygous dominant genotype, like big A, or it could have the heterozygous genotype, like big A, little a. Either way, that parent would show the dominant physical trait. So if we're trying to figure out which of these two genotypes that dominant parent has, we can cross that parent with a second parent that has the recessive phenotype. Why do we do that? Well, we know that second parent can only have the homozygous recessive genotype, little a, if they're going to show the recessive phenotype. Normally, when we do a Punnett square problem, we have information about the parent genotypes, and we're trying to figure out information about our offspring, our predicted offspring genotype and phenotype frequencies. In a test cross, we're kind of working backwards. We have some information about our offspring, and we need to find out some information about the genotype of the parent who has the dominant phenotype. So instead of going from parents to offspring, we go from offspring to parents. Okay, so we have two possible results from our test cross. Okay, so let's say all of the offspring show the dominant phenotype. I've set up kind of a working backwards Punnett square on the right here. Remember, one of our parents we know is homozygous recessive, and we're just trying to figure out the genotype of the second parent. So if all of our offspring show the dominant phenotype, that means that this parent, the dominant parent, had to have big A as their genotype or homozygous dominant. So let me go ahead and fill that in. And notice how that will result in all of the offspring showing the dominant phenotype because they're all heterozygous for the trait. So in this case, the missing parent genotype was big A. Alternately, the results of your test cross could be that half of your offspring show the dominant phenotype and half of them show the recessive phenotype. So we could see this in our Punnett square that we're working backwards from here. We know one of our parents, again, is little a, and we could have half the offspring show the recessive phenotype and half show the dominant phenotype if they have these combinations of alleles. That would only happen if our dominant parent was heterozygous, big A, little a. So the missing parent genotype in that case is big A, little a. So again, if all of your offspring show the dominant trait, that means the parent was homozygous dominant, big A. And if half of your offspring show the dominant trait and half show the recessive trait, that means the parent was big A, little a, heterozygous. Okay, now thus far we've only looked at traits where you have one allele that totally covers up the expression of the other allele. One of them is dominant, one of them is recessive. These are called traits that display complete dominance or simple dominance. Okay. And in this case, if an individual has the heterozygous genotype big A, little a, they're always going to show the dominant physical trait. Now, we're going to look at two examples where this is not the case. The first is called incomplete dominance. And incomplete dominance is when the organism that has two different alleles, so a heterozygote, is going to show a phenotype or physical trait that's a blend of the two alleles, so somewhere in between the two alleles. Because of that, we often call this form of inheritance blending inheritance. So let's look at an example. So here we've got some snapdragons, and we've got two parents. One of them has red flowers, one of them has white flowers. And when you cross the two parents, all of their offspring are pink. So they have an intermediate phenotype between the two parents, red and white. Another example would be if we had two alleles, one for short stems and one for long stems. And if you get both of them, you have a medium length stem. So a blend of the two, intermediate between the two. Okay. Now, usually with cases of incomplete dominance, we're going to use capital letters to represent both alleles to show that neither one is dominant over the other. Like we might use a big R for red flowers and a big W for white flowers. Sometimes you'll see examples where you have one uppercase letter and one lowercase letter, but I don't really like that system because it does imply that one is dominant over the other, which is not the case. So here's an example with our red flowered and white flowered parents. So to have red flowers, you need to have 2 copies of the red allele, and to have white flowers, you need to have 2 copies of the white allele. We would call these parents purebred, which means they're homozygous. They each have 2 of the same allele. And when we set up a Punnett square between these 2 parents, all of our offspring have the genotype RW. And in this case, that means that they have pink flowers. And I would have to tell you that prior to you completing a Punnett square, that this is a case of incomplete dominance and any heterozygote has pink flowers. So I would have to let you know that before you did the problem. Okay. Our next type of trait that does not show complete dominance is codominance. This is when organisms that have a heterozygous genotype, so 2 different alleles, show a phenotype that displays both of those alleles in their pure form. So they both show up independently and equally in the phenotype. So for example, let's say you have 2 parents that are that have red and white flowers in this camellia plant. That will result in offspring that have flowers with patches of red and white. So it's not a blend of the 2 parents. Both parents' alleles, the red and white alleles, are showing up in their pure form. Okay. Another example might be if you had some aliens and there was one allele for curly hair and one for straight hair. That would result in an offspring that has some hair strands that are totally straight and some that are totally curly, so that both of those alleles are showing up in their pure form. Okay, so let's set up an example Punnett square for 2 aliens that are heterozygous for hair texture. So I'm going to go ahead and put a little table to represent my Punnett square in here. Okay. No, I don't want to leave. Cancel. Okay. All right. And if those 2 aliens are heterozygous for hair texture, what that means is they have 2 different alleles, so they're each going to have a genotype CS. We want to write the first letter that comes in the alphabet first, so we'll start with the C and then the S for their genotypes. Okay. And I'm going to go ahead and put the alleles for both of those parents on the 2 sides of my Punnett square. First parent goes on the top, C. Switch bigger. And okay, so we have our first parent's genotype, CS on the top. And our second parent's genotype is is the same. So CS on the left side. All right, and that is going to result in offspring that have the following genotypes. This one right here will be CC, this one will be CS, this one will also be CS, and this one will be SS. So 25% or a quarter of their children are going to have totally curly hair. 50%, so 2 out of their 4 potential offspring, will have a mixture. So they'll have some hairs that are curly mixed in with some hairs that are straight. And then 1 out of the 4 offspring, or 25%, are going to have the genotype SS for totally straight hair. Okay, that's it for this video. Thank you.